myself. Yeah, so I want to talk a little bit about a group of spiders that I spent a lot of my career studying. And it's a very speciose group, which actually makes up a significant fraction of Newfoundland's biodiversity, whether people are aware of it or not. Before I start, I'd like to thank once again Cole Deal and Linyphiidae for helping me get this set up and everything. And especially I'd like to thank Michaela Swain, who was a student at UW Green Bay, for basically suggesting me for this talk. And so if it wasn't for her, I wouldn't be here. So I really appreciate her support. Thank you, Michaela. All right, so my outline. First of all, I just want to talk about what sheet web spiders are and sort of place them onto the tree of life. And then I want to try to explain why would someone want to spend their career studying Linyphiids. We don't actually use the term Linyphiidologist, but I think it's funny, so sometimes I call myself that. But I especially want to talk about this really important thing that has to do with invertebrate biology in general, which is a sort of a taxonomic impediment to doing research on these organisms. And that's really what stimulates my interest, at least at the beginning in this group. And then I want to end up by asking, not really answering, but just asking some big questions that we still have about this lineage of spiders. And it really was partly inspired by a kind of a friendly argument that I had with another arachnologist, Ingi Agnarsson, an Icelander who's now at the University of Vermont. And he was basically asking me, what's so special about Linyphiids? And so I want to try to answer that question. I'm going to sort of mention my research sort of tangentially as we go through this talk, but you know, a taxonomist's research doesn't really form a real story narrative. You know, it's like you stumble upon things, you discover things, what you end up working on is somewhat random. And so the real story here is kind of what I've learned about Linyphiids from studying this taxon and just from everybody studying this taxon. So first of all, a little bit about what sheet web spiders are. So I want to kind of focus on where spiders are on the tree of life. I think this is really important. The tree of life is really the most profound discovery that taxonomists have contributed to human knowledge. You know, this idea that all species on the planet are all related to each other, and every species has a common ancestor with every other species. It's a really profound idea that changes our outlook on the planet. And we actually do share common ancestry with Linyphiids, and we're in the same group as them at the beginning, right? We're both, you know, eukaryotes, just like all multicellular organisms, and we're actually humans and spiders are in the same group that includes fungi, the opisthecons. We're both animals, of course. Kingdom Animalia is a monophyletic group that includes all the moving, pooping organisms on the planet. And then when you get to the phylum level, that's when we sort of part ways, right? So obviously we're chordates. The spiders are in the phylum Arthropoda, these animals that have a chitinous exoskeleton, and so they have jointed appendages, which are so useful to them. The arthropods are broken down into several big groups, and the spiders are in the subphylum Chelicerata. And these are animals that don't have jaws like insects and crustaceans. They have either fangs or pincers instead of jaws. And of course, they also lack antennae, which you kind of associate with insects and crustaceans and so on. The most famous chelicerates are the arachnids, the most diverse group of chelicerates now. They're the ones, of course, that have 8 legs, and includes scorpions, mites and ticks, as well as the spiders. So finally, we get to this big order of arachnids, the order Draney. That's the spiders, and it's definitely a monophyletic group, right? A natural group that has one common ancestor. And there are a number of really interesting traits that are shared with, they're synapomorphies. They're shared derived traits that are unique to the group. So all spiders have these traits, and no non-spiders have these traits. So I want to point out too, can you see my arrow? You can see my arrow, all right? Huh? All right, so on the back end of spiders, especially on this spider, you can see these kind of exhaust pipes, and these are the appendages that make the silk, right? These abdominal silk-producing appendages are called spinnerets, and all spiders have spinnerets, and they make silk, whether they make webs or not, they all make silk. And these spinnerets are a synapomorphy of spiders. The other synapomorphy of spiders that's really interesting is the way that they mate. Spiders, their sperm transfer organ is actually modified mouth parts. So the males, the second appendage of their mouth parts, the pedipalps, they look like little short legs. In male spiders, they sort of look like boxing gloves. And this is actually the intromittent organ that transfers sperm to the female. So this method of mating is really weird, and all spiders do it and no other animals do it. So that's a couple of things about spiders that I think everyone should kind of know about. Now let's talk about the Linyphiids and where they are with regard to the spiders. And this is a kind of a pattern that you see when you look at big groups of organisms of any, clade, and that is really diverse groups tend to be in really diverse parts of other diverse groups, which are themselves parts of other diverse groups. And so what you're seeing is animals, in this case, that have inherited some real evolutionary advantages from their ancestors, which have allowed them to really speciate and to become incredibly successful on the planet. And so obviously spiders are in the arthropods. This is the largest phylum of organisms, period. And about 3/4 of all described animal species are arthropods. So it is by far the largest animal phylum. The spiders are a pretty big group themselves. So they're the second largest order of arachnids, the biggest one being mites and ticks. So there's over 48,000 species of spiders described so far. So spiders are divided into two suborders. The mesothelials are primitive spiders that you can still see some evidence of abdominal segmentation on their abdomen. And then that's actually a pretty small group, one family and about 138 species. So less than 1% of all spiders are mesothelials. Most spiders, including Linyphiids, are opisthothelials, and that includes 127 families and more than 99% of all spiders. Okay, so then this suborder is divided into two infraorders. There's the megalomorphs, the tarantulas and their kin, and then there's the araneomorphs, which are the kind of spidery looking spiders that you're probably more familiar with from Newfoundland. So the megalomorphs are pretty cool, and it's a reasonably diverse group, but so there's 30 families and over 2,000 species, but this is only a little bit more than 5% of all spiders. So almost 95%, 94% of all spiders right now are in the araneomorphs. That's 97 families. And then when you look at the araneomorphs, they're divided into a number of really big groups or clades. And one of the biggest ones is the superfamily that Linyphiids are in, the araneoids. The araneoids include the orb-weaving spiders that make that spiral web that you guys are probably familiar with, and then a bunch of other families. So there's 17 families of araneoids, and they include about 12,000 different species. And so it's a very successful group. 13% of all spider families make up over 1/4 of all the species. So a very successful group of spiders, web-spinning spiders mostly. And the biggest group within the araneoids is the family Linyphiidae. So this is the largest family of araneoids, about 37% of all araneoid species. So there's a picture of a Linyphiid spider. So we are starting to really understand, arachnologists, biologists, starting to really understand the phylogeny of this really amazing group of animals. And it's really given us the power to understand them in a whole new way. So this is one recent phylogeny that's come out, a really beautiful one here, I think, in 2016. And you can see right at the bottom of the clade here, right, in a really derived group is the araneoids. And so those are the orb-weaving spiders that are related to Linyphiids. And then there's the Linyphiids right there nested within the araneoids. Spiders themselves are a really ancient group, right? They emerged way before the dinosaurs in the Paleozoic, the Devonian period. So that's over 380 million years ago. Linyphiids are a considerably younger group. They emerged sometime in the age of the dinosaurs. The oldest Linyphiid fossils that we have are 125 to 135 million year old fossils from amber from Lebanon, and these are from the lower Cretaceous. So it is quite an ancient lineage too that evolved during the age of the dinosaurs. So family Linyphiidae, it's hard to say and it's hard to spell. We usually just say sheet web spiders. The name actually is Greek. Linon means a line or thread, and phia means maker or builder, something like that. So they're the line weavers or thread weavers. Although most of the time we just call them sheet web weavers because they make this special web that's sort of unique to the family. They do share some important traits with other araneoids. They are 3-clawed web spiders. A lot of spiders have just 2 claws, but web spiders have a 3rd claw that allows them to kind of grab a silk line more readily and walk on their webs more efficiently. And then they also are intelligine spiders, which means they have very complicated, complex genitalia, which is really fortunate because that's mostly how we identify them, by the differences in genitalia. Linyphiids are famous for this non-sticky sort of horizontal sheet web. The photo I'm showing here is a really big one that photographs well. This is the filmy dome spider, and the sheet is sort of drawn up into this dome, but a lot of them just have a horizontal sheet. The spider hangs out upside down underneath the sheet. So this is the one that made that web there, Draney. Litigiosa, a big spider that's pretty easy to photograph. So they forage upside down underneath this non-sticky horizontal sheet web. And then there's a bunch of scaffolding above and below the sheet. This knocks down insects into the web, and it also protects the spider from predators and parasites. It's not just a capturing device, but also a protective device for the spider. These spiders have relatively thin, spiny legs. And one interesting thing about them is they detach at the knee joint, sort of midway down the appendage, rather than at the shoulder, at the base of the appendage, like most spiders do. If you grab onto a spider's leg, it can amputate its own leg, and Linyphiids amputate at the knee, which is an unusual trait. One interesting thing that I'll talk about a little bit later is that a lot of Linyphiids have modified carapace or head region of the males. Just the males, not the females, have these modified head regions. And that's one of the reasons why I got interested in this group in the first place, because they're just really cool looking. They're kind of alien looking. They are really noted among arachnologists as being really tiny. They're among the smallest spiders. There are other small spiders, but they get down to less than 1 millimeter in body length. And the average is like 2 or 3 millimeters, right? They're rarely over 1 centimeter. So the vast majority of Linyphiids are too small to bite humans. So this is not like a macho sort of field, you know, like studying tarantulas or alligators or something like that, you know, but they're still amazing animals. And of course, there's a lot of other genital characteristics that separate Linyphiids from closely related spiders. They're actually hard to identify. It's hard to tell them from other web spinning spiders. So a little bit about the ecology of Linyphiids. I sort of think of them as the premier web building spiders that build their webs in leaf litter. That's the main habitat for most of the species, and they are found all around the world in leaf litter. They mostly make really small webs, a couple of square centimeters, maybe the size of a postage stamp. And so they're just a couple millimeters. They make their webs in the interstices, or the spaces between leaf litter. And that's the main habitat, but you can definitely find a lot of Linyphiids in moss, in wetlands, peat moss, sphagnum moss. Many species are found in vegetation also, like we see in this photo on the right here. Some of them even occur in the canopy and tree trunks of forests. A lot of species live on bare ground in agroecosystems, or up in the high tundra or in deserts, sometimes underneath rocks for shelter. Some species have evolved the ability to live inside of ant nests and are commensals with ants. And then a good number of species have evolved to life in caves, right? So they're often, you know, in the Northern Hemisphere, they're often spring reproducers, that they overwinter as immatures. They become mature in the spring, and that's when they reproduce. They usually have 1 generation per year, like say up in Newfoundland, but more species become multivoltine with many generations per year as you go to warmer and warmer climates. And they're also famous, Linyphiids are famous for ballooning, right? Aerial dispersal on silken lines. And they oftentimes do this in the spring and the autumn, depending on the species. And this is one of the very few spider species that can balloon even as an adult. So they're really great at dispersal by ballooning. One of the secrets to their success. Different groups of spiders have different mating positions, you know, and so I thought I'd show you up there at the upper left, the mating position of Linyphiid spiders. They hang upside down in their webs, not surprisingly, with the male sort of spatially above the female, but anatomically below the female. They're both upside down. So it's kind of an interesting, unique mating position among spiders. So a little bit about spider sex, right? We got to talk about these modified carapaces. And this is a great example of sexual dimorphism in spiders. These are the carapaces. So the legs have been removed and the abdomens have been removed from these drawings. So you can just see the carapaces. They're all facing to the left there in the screen. And these 4 are all in one genus of Linyphiidae. So these 6 are all from 6 different genera, but and this same genus has males that have unmodified carapaces too. So although they can make the animals fairly easy to identify to the species level, it's actually made the genus level taxonomy more complicated. Because it doesn't really map onto the actual natural groups. So this is a great example of sexual dimorphism that's not size dimorphism, where the famous kind of dimorphism among spiders is that males tend to be a lot smaller than females in many spider groups. But that's not the case with Linyphiids. Male Linyphiids are about the same size as females. And in fact, in some species, the males are a little bit bigger than the females, possibly because of competition between males, male competition might make them a little bit bigger. And so that's kind of an interesting thing among spiders that's not very common. So here's a case. I want to tell you a story about a polymorphic species of Linyphiidae that I think sheds some light on why they have these modified carapaces. So it used to be in Western Europe that people thought there were these 2 species of Oedothorax. There was Oedothorax gibbosus, which has this kind of hairy armpit-like structure on its head. And then there's one with a regular carapace for a spider. It's called Oedothorax tuberosus. And people thought those were 2 different species until a very simple and devastating experiment proved that to be wrong. People were rearing spiders out of a single egg sac, and they would get both kinds of males. So they were brothers, they were siblings. And so it was a polymorphic species. And so people started studying this, and they realized that the females really like the gibbosus, the modified form. Apparently, this male produces a sexual pheromone that gets trapped in this pit here, and females will actually insert their fangs into the opening there and sort of try to take in this substance. So these males are very sexy to the females, and they have much greater reproductive success. And then, of course, you'd ask, well, why are there the unmodified ones? Well, during wet years in Western Europe, a lot of the times these modified males, they get fungal infections inside of this pit, and they die. And so they tend to be dying off, and the ones with the unmodified carapaces are surviving, and so they have much higher reproductive success just because they survived during those years. So it's an interesting case of sort of a balancing selection which maintains both of these morphs in the population, and it really shows that sexual selection, basically female choice, is really what's maintaining this interesting morphology in these populations. So let's talk a little bit too about the evolution of the web, this Linyphiid sheet web. Now that we are starting to have a really good idea of how spiders are related to each other, we're starting to be able to ask and answer really interesting questions like how the webs evolve. It's really interesting because it's a non-living thing, but it's a sort of a record of the behavior of the spider. And we used to think that, so there's a whole bunch of different groups that make orb webs, and orb webs are a really amazing structure, not much silk, extremely strong, easy to make. And biologists always thought this was the pinnacle of spider web evolution. And it was believed that very unrelated species evolved the orb web independently several times because it was just like the engineering marvel. Now that we've gotten a pretty good idea of the phylogeny, though, it seems very clear that all of these orb web spiders all inherited the orb web from one ancestor, okay? And then after that, these orb webs were lost many times and modified quite often too. So this big group of 2-clawed spiders that includes jumping spiders and wolf spiders, they seem to be descended from orb weaving ancestors. And same with the sheet web. It used to be thought that the sheet web was like a primitive web which led to the orb web eventually. But in fact, it's the opposite. The sheet web is a highly modified orb web. And so the orb web is not the pinnacle of spider evolution. And actually, the sheet web is descended from it, you might say. So anyway, I don't have time to talk more about it, but that's a pretty interesting story. So that tells a little bit about Linyphiidae. Now I want to talk a little bit about why someone would be wanting to spend their entire career studying this group of small spiders, and it has a lot to do with this taxonomic impediment. And I want to talk a little bit about my career too, to give you an idea of how I came to be an arachnologist. So I grew up in New Mexico, and it's a great place to become interested in arthropods. There's all kinds of cool spiders and insects and so on. But probably an even more important factor in me becoming an entomologist is just that I'm extremely nearsighted. And so like as a teenager, I was very interested in birds. But you know what? I'm a terrible birder. I can't see the birds very good. I can't tell them apart. I'm a terrible hunter too. You know, I gave up on that really quick. But I have a superpower because of my nearsightedness. When I take off my glasses, small objects are magnified greatly. And so it's really useful for studying insects and spiders. And I played with them all the time when I was a kid. So I'm going to show you a photo of me when I was younger. This is actually the oldest Mike Draney I could find that's not wearing glasses in the photograph. And so there I am. This is from, I think, Christmas of 1973 when I was 6 years old. So after that, every photo after that is showing me with really thick glasses. And so that had a lot to do with me growing up being really interested in insects and spiders and stuff. I kind of thought that I gave up insects and spiders when I went to university. So I went to the local state university and I thought, I'm going to become an ecologist. I'm going to study plant ecology because plants are at the base of the food web and they're super important. Okay. And, you know, I like plants. I still, you know, am interested in plants. But by the time I was a senior, I realized, you know, those insects I was playing with are extremely important ecologically. And actually, there's a lot of jobs for entomologists. So I was working for a biologist who is an invertebrate biologist, Dr. Marcia Conley, and I wanted to do a senior thesis project. So I asked her, "What do you have some research I could help with that you know, that I could do a senior thesis project with?" And she said, "Yeah, you can either work on harvester ants or you can do this project on black widow spiders." And so I had my choice of these two red and black animals. Pretty cool. And I thought to myself, you know, I've already played with harvester ants when I was a little kid. I always had ant farms and things like that. So I want to try something new. So let's study black widows. And so what we were looking at is that every time you flip a cow patty in New Mexico, there's a black widow under there. And so my major advisor said, "I wonder if you had more cow patties, would you have more black widows?" So we tried to do an experiment to test that. And it turned out to be really hard to test, but I got really interested in spiders by doing that. So then I went to graduate school and I thought, once again, I was going to study something important. I said, I'm going to study mites. They're even more diverse than spiders. And I'm going to study soil ecology with the world famous soil ecologist. So here he is, D.A. Crossley Jr., everyone calls him Dak. And so I went to the lab of Dak Crossley. He was actually a chigger expert as a young man and then turned into a soil ecologist. But he was very prominent, very productive, lots of graduate students, postdocs, and I was the youngest one. Let's face it, I was just being ignored in his lab, okay? And so I started doing what I knew how to do already, which was I was catching spiders and trying to learn the spiders in Georgia, 2,000 miles away from where I grew up. And so he saw me doing that one day and he said, "Why don't you study spiders for your master's degree? And then when you do your PhD, you can do mites." I said, "Okay, great." And I got so interested in spiders as a master's student that I never went back to mites. And when I was a master's student is when I discovered this taxonomic impediment. I was trying to study spider ecology, but it turned out that I spent most of my time trying to figure out what spiders were which. You know, you have to have the name so you can put them in the right groups and so you can calculate diversity and so on and so forth. And so I almost became a spider taxonomist like a lot of spider ecologists do. And so this taxonomic impediment is really important for invertebrates, and it limits the amount of research and the amount that we know about them. So let's just consider spiders as a whole, right? It's this awesome lineage, but they can be very difficult to study and to identify the species. They're the 7th largest group of animals. So there's 5 orders of insects, and plus mites and ticks have more species. But after that, they're the biggest group of animals, over 48,000 species. They're the largest group of animals that are entirely predators. So that's pretty interesting. You know, there are other groups like, I don't know, sharks maybe, dragonflies and damselflies is entirely made of predators, but this is by far the largest group of organisms that's all made of predators. And I was really interested in them too, because that you can find spiders everywhere that humans live, right? So it doesn't matter where I go, there's always going to be spiders to study. They're a pretty prominent and usually the major predators in pretty much every terrestrial and semi-terrestrial habitat, except for Antarctica. So other than Antarctica and the ocean, you're going to find a lot of spiders. So they're found in intertidal zones. They're found in freshwater margins. They're found in all types of wetlands, from the driest deserts to the wettest rainforests to high Arctic tundra, as high up as you can go. And they're also, interestingly, spiders are the highest elevation animal that has ever been discovered. They live on the slopes of Mount Everest, 6,700 meters up, a couple of 100 meters above the highest, marmot or yak or whatever they've ever found. And you're probably asking, what is a predator doing up there by itself? Well, insects are dying up in the sky, and then they rain down on the slopes of Mount Everest, and they mostly seem to eat dead bugs. So this is it. This photo here is a jumping spider that is the highest elevation jumping spider, Euophrys omnisuperstes, which is a great name. It means I am above all. Pretty cool. All right, so they're awesome, but they're difficult to identify. And some of the reasons why spiders are difficult to work with is the same things we have for all sorts of animals. They're a lot of species, and many of those species are actually even undescribed, right? Or not very well described, especially as you move into the tropics. These spiders are mostly really small animals, and the Linyphiids certainly are quite cryptic. So, if I really want to study, Linyphiids, oftentimes I get little handfuls of leaf litter and put them into a bag, and then take the bag back to the lab and very carefully sort through that to try to find them. That's what I'm doing there. That's me in Panama with the bag of leaf litter I was really happy to get from Barrow, Colorado Island. So yes, this is a quote from my colleague, Mark Milne, which I think pretty well sums up our lives with spiders. Some of the problems with spiders, though, go beyond just invertebrate problems. Spiders have their own problems, which make them particularly difficult to work with. And one thing is, unlike butterflies or beetles or something like that, you can't just look at a photo of a spider and know what species you're looking at, right? So site ID and photo ID is often impossible, because we have to look at the genitalia of spiders, often at a really high power under a microscope. They do sometimes have different coloration and markings, but oftentimes they're highly variable even within a species. So they can actually be misleading, right, and fool you. Basically, Linyphiids pretty much all look like this one, That's the thing. So if you see this, even in a world expert, we've got some of the world experts here in this room, and who knows what genus that is? I don't know. I don't know what it is. Another frustration is that immature spiders are usually not identifiable, for the reason that we have to use mature genitalia, and they don't have those yet. The immatures can be very ecologically important, so that's very frustrating. The adults of spiders are often not present year-round. So this is the phenogram of one species of Linyphiid, and you can see mainly the adults are found November to April. So if you were looking for this species in July or August or September, you're just not going to find it. So you can't just go to a site and know what spiders live there. You have to go several different times during the year. And then the real kicker is that, the male genitalia are mouth parts, the female genitalia are in the abdomen. And so they're very different character systems. They're completely different character systems. And so essentially, when you learn spiders, you're learning two different sets. So if you think you're going to learn 1,000 species, you're really learning 2,000 different forms. And so there's twice as much to learn. Females are often more difficult to identify. My colleague Nina Sandlin here today is kind of an expert at the female parts. They just don't vary as much as the male genitalia. So it's often hard to tell the difference. And then, of course, we have this problem of gender matching, right? So we think we know which males and which females to go together, but we usually haven't seen them mate or anything. So this is actually 3 different species of Linyphiids, and the males are on the top and the females are on the bottom here. So, you find a male and a female, and you might think that they go together, but a lot of times people are wrong, right? And so that is an extra complication that we face. So this is just an example of how difficult the identifications can be. one genus of Linyphiids, Agonita. There's about 200 or so species around the world. And I'm just showing one photo because most of them look pretty much like this, right? There's a little bit of variation, but you definitely cannot identify them by just looking at photos. We have to very carefully study the genitalia. This is the male genitalia on the top here, and then the female genitalia is towards the bottom here. And this is a species, by the way, that's named after my mentor, Don Buckle, so Agonita buckeli. And then this is just another species for comparison. And you can see, well, for the male, it's pretty easy to separate the males. If you look at that symbial knob there, that's pretty distinctive. But the females, they're pretty similar, and it can be really hard to identify them even under the microscope, let alone, with a photo. So this leads to what I sometimes call the BioBlitz problem. It basically means that arachnologists have to be very patient because you're not going to learn things right away. I go to these BioBlitzes, and there's all these different biologists, and they're all looking at their special group, and we're trying to figure out what plants and animals live in one place at one time, right? And so the botanists, I'm really jealous of the botanists. They walk around usually with a clipboard with a checklist, and they're just checking off plants just as fast as they can. A lot of times they don't even have to bend down to look at it, right? It's like it's a maple tree, but it's obvious. Every once in a while they'll bend down and look at one in a magnifying glass or something. But so at the end of the day, they'll be like, hey, Mike Draney, I just found 313 species of plants. How many spiders did you find? And yeah, Mark knows where I'm going with this. I always tell him, I have no idea how many spiders I found. I'll let you know in about 2 weeks. And then I go to the lab and I spend many, many hours behind the microscope trying to figure out what it is that I collected that day. So you have to be very patient. It can take weeks, months, even years to figure out what you have. So that slows down research considerably. It is very much of a frustration. And just to show you what a big group, spiders tend to be, I thought I'd introduce you to the biggest, most diverse groups of spiders. And first of all, number one is actually these really cute spiders. This is the most common type of spider on the planet. These really cute, short-legged, big-eyed, jumping spiders, diurnal, very smart, very active, curious. They're mostly tropical, but they're found in Newfoundland. All 7 of these are found in Newfoundland. This is the biggest group of spiders in the world, over 6,000 species. So over 12% of all spiders are jumping spiders. And then there's Linyphiidae. That's the number 2 most diverse, most speciose group of spiders. Over 4,000 species and over 600 genera. So it's almost 10% of all spiders are Linyphiids. That's Gonatium crassipalpum, which is found in Newfoundland. Here is a spider you might be familiar with. That's the cross spider, a European garden spider. And that's an orb weaving spider, right? So it's number 3, 3,000 species, about 6% of all spiders. And then we have a spider you may not be familiar with. This is the ground spider or running spider. And this is Zelotes fratrus, which can be found in Newfoundland. And there's about 5% of all spiders are these, over 2,500 species. Same with the cobweb spiders, the Therideids. That's Steatoda bipunctata. It might live in houses in Newfoundland. I don't know. And then let's see, we've got, wolf spiders. Most of you are familiar with wolf spiders. This is Pardosa moesta, a spider that you can find in like wet meadows in Newfoundland, I'm sure. And so again, about 5% of all spiders. And then finally, we have the crab spiders. This is the flower crab spider, Mycemena vatia, which occurs in Newfoundland. And it's a great spider with that sexual dimorphism, that huge white female, camouflage, and that little tiny dark male sitting on her abdomen. So those are some really big groups. I don't know of anyone who really knows jumping spiders and sheet web spiders at the same time. There's just too many species, and so you have to specialize if you're going to identify spiders to the species level. So when I first started working on spiders, this was the book that we used to identify spiders, Spider Genera of North America. And it has a chapter for every family of spiders, and each chapter has a key to that family, which takes you to genus level, right? So you can key out all the genera of spiders in North America, and every family has a key except for one, the Linyphiids, right? And if you go to the chapter on Linyphiids, this is what you see instead of a key. You see this big alphabetical list of over 150 genera, and it says what states and provinces that spider has been found in, and it has some papers that you can go look up, and then good luck, man. That's it. That's all you had. And so a lot of my career has actually been trying to help people be able to identify spiders, you know, and this is one of the reasons I really got interested in them too and decided to work on it for my PhD. Is just, it's like a challenge. It's like, oh, I want to be the guy that can do this, that can crack this puzzle. And I also want to make it a little bit easier to do. It took me a long time, a lot of sweat to learn how to identify these, and I'm trying to make it easier for the next, you know, generation. So arachnologists decided to update this book, and they came out with Spiders of North America, an identification manual. And I told the editors, hey, I'm going to make a key to the Linyphiids. Is that cool? And I would have never been able to do this if it wasn't for my colleagues. So this is Don Buckle. He is a Canadian, so I thought I'd mention him. He's also my mentor, my Linyphiid guru. What he's doing there is he's writing his outhouse in northern Saskatchewan. It was frost heaved over the winter. So I don't know if you guys are familiar with doing that kind of stuff, but there he is. And we basically got together and we talked about how we were going to split this up. We split the family up by subfamily. He took one, I took the other. Then we met for a week in Saskatoon. By the way, Don is by profession a locksmith in Saskatoon, Saskatchewan. That's what he does. And then he spends his winter nights, you know, up in his attic, and he's one of the world experts of sheet web spiders. That's a cool thing about spiders too, is that a lot of amateurs, you know, that word maybe sounds dirty to scientists, but the amateurs have contributed a lot to spider knowledge. So we came out with this chapter 35. This is probably one of the things I'm most proud of in my career is this key to 157 genera and almost 1,000 species of Linyphiids in North America. And it has a key. So this is one of the pages of the key, lavishly illustrated by another Canadian, a Quebecois, Nadine Duparé, did the illustrations. And let's face it, it's a difficult key to use. These animals are still difficult to identify, but I'm really proud of doing this and allowing people to get into Linyphiids a little bit easier than I did. So that's what a lot of my career has been about, really, is about helping other people work with spiders. And I'm interested in ecology. I'm an ecologist, but a lot of the work I do, I end up collaborating with other people. So I'm interested in documenting biodiversity, especially in a conservation context. And I'm interested in finding rare and threatened taxa. And I'm also very interested in using spider communities as sort of indicators of change, right? Whether it's big changes like invasive species or global climate change, or whether you're talking about management strategies and how that changes the spider communities. A lot of this work I've done in collaboration with graduate students. The graduate students do most of the field work. They're mostly interested in the ecology and conservation aspects, and then I help them to identify the spiders to species so that we can do good work. And then a lot of times I'll also end up being kind of a subcontractor, sort of an identification subcontractor for ecologists. Ecologists get really into trapping spiders and catching them, and they get really interested in them. And then they get back to the lab and they realize, oh geez, this was worse than I thought. This is going to take me years to figure this out. And so then they email me and say, hey, can you identify these for me? And so I get the spiders. And so that's how I've done a lot of work, actually, as just in collaboration with ecologists. So I'm just going to show you some maps that show some of the places I've worked on. And so this is the Western Great Lakes region. There's UW Green Bay right there at the mouth of the Bay of Green Bay, the biggest freshwater bay in the world. And you can see that I've kind of taken this side of the Great Lakes and done a lot of work there with collaborators. The embarrassing part about these maps is that these red dots are projects that have not been published yet. So there's going to be a lot of red dots. The green ones are ones that I've published, and then the yellow ones are ones where we've published some, but not all of the information so far. So you can see myself and collaborators have done a lot of work in Wisconsin and tried to improve our knowledge of Wisconsin. I've done work around North America, though. I had to really zoom out so we could get northern Alaska here. So I've done some work, again, as a subcontractor up at Toolik Lake, long-term ecological research site in northern Alaska. And so I've published 2 papers on the spiders of Alaska, and I've never been there before. So I don't know if that's sad or amazing, but anyway, it allows you to kind of understand places without ever having visited them. But I've done work in New Mexico, where I'm from, Colorado, Kansas, down in South Florida. This is where I did my graduate work, right? Georgia and South Carolina. I did some soybean work up in Virginia. And then this is actually the closest I've gotten to you guys. other than maybe Iceland, is Nantucket Island, which is the island off the coast of Massachusetts. And one of my graduate students was working there and did his thesis research on that island. I've become, in the last decade or so, I've become very interested in the spiders of the northern neotropics, kind of where the neotropical spiders meet the Nearctic spiders in Central America and the Caribbean. I'm really interested in this area, and so I've done some work in the Keys and in the Everglades, as well as down here in Trinidad, a little island called Dominica. Here's a coffee plantation in Mexico, and then I've sampled a bunch of spots in Central America, and that's kind of a project I'm going to be working on probably until I retire. And that I'm going to zoom in on that finally for the last map here. So this is just showing some of the sites that I've been to in Panama and Costa Rica. Since 2007, I've been taking students on these 2-week trips down to Panama and Costa Rica to show them around. And one of the things we do is we look for spiders, right? So I've got really good data on all these spots, and it's been really a satisfying project in a lot of different ways. It's fun to go to the tropics. Of course, there's a lot of new species there. It's great for the students. It's a great experience for the students. It's very good for me too, though, because 10 students can collect more spiders in a day than I can collect in a week, right? And so it's very efficient. And what we've been basically doing is trying to develop a rapid assessment protocol. So, people go to the tropics and they collect here and they collect there and they say, I think this place has the most spiders. But, they collect different numbers of spiders for different amounts of time using different methods. And you just don't really know which spot has the most spiders. So what we're doing is we do the exact same kind of collecting for the exact same amount of time everywhere we go. And then hopefully we'll be able to, rigorously, statistically compare which sites really do have more species and so on and which don't. So this has been a really fun, satisfying project, introducing, a lot of students to this work. So the thing that I'm really the most satisfied though in my career is finding and documenting new biodiversity. And a lot of times this stuff happens as a result, sort of a byproduct of doing ecological work. People discover species, they're not really looking for them, but they're part of the catch. And so we find new species, or sometimes we just find new biogeographic information. We find species where they've never been found before, and those can really extend our knowledge of where spiders live. And this is just one example. This is a drawing by my wife of a species that I sort of discovered, this new species of Linyphiidae when I was doing my PhD work. And this was 25 years ago, and I couldn't figure out what it was. I couldn't even figure out what genus it should be in until I started working with Dr. Mark Milne from the University of Indianapolis. And we finally think we figured out what genus it should be in. And then he did good illustrations. We described this species. We revised another species. And that was a real blast to finally get that out into the world. Really beautiful spider. I should say it's named after a former colleague, a late colleague of ours, Mandy Howe. And so we were both really happy to get that out there. So this is one advantage of studying a difficult and diverse taxon, right? There's a lot of disadvantages, but one of the advantages is it's pretty easy to make new discoveries, right? And so I just want to show you one here. It's teasing you a little bit, but I found not only just new state records, spiders had never been found in Wisconsin, but this is a spider that had never been found in the United States before. But what's sort of funny about it is Canadians have a list of spiders they feel are endemic to Canada, right? These are Canadian spiders, right? Well, this was formerly a Canadian endemic spider. It was only known from Manitoba, New Brunswick, Ontario, and Quebec until I came along, and I was actually doing work in the city limits of Chicago, and found this little 1.5 millimeter long spider, this is the southernmost record of it, of course, on the planet. But a new national record, and it was just a real gas. And of course, the Chicago papers were interested in it too. And then people started reporting it in airports and stuff, which is another fun thing about working with spiders. Everyone knows about spiders, so you don't have to explain yourself too much. Sometimes it goes beyond just putting a dot on a map. Sometimes we can really learn a lot about spider biology just by collecting them in new places. So here's an example. A colleague was trapping animals in pocket gophers in the upper Midwest. And he sent me the spiders to identify, and one of them really freaked me out. This one looks like a cave-dwelling spider. It's got tiny little eyes, and it didn't really take me that long to figure out what it was because it's got these big hairs on its genitalia that tells me that it's in an Islandiana, in the genus Islandiana. And then you go to the book, and it's the one jumps out at you. There's one Islandiana that's got this big horn on the top of its head, right? It's a modified Seta. It's the unicorn Islandiana. And so, easy to identify. But I was like, this can't be, because the only place this has ever been found is in a cave in North Texas. And so that was really puzzling that we found them in pocket gopher burrows in the Midwest. But when you map the distribution of this species to the distribution of pocket gophers, things become a little bit clearer. I later learned that this species had also been found in caves in Missouri. And both the Missouri caves and the North Texas cave are both within the range. This polygon here is the range of pocket gophers. There's different subspecies or something. And so these are the places where we found them, you know, in the upper Midwest. And so probably this species occurs wherever the pocket gophers are. And then occasionally they find a way into a cave from there. And this is probably, you know, otherwise it's a mystery how this spider gets from North Texas to Missouri or back. But now we think we know how it moves around. It lives in pocket gopher burrows. So that's pretty cool. So this is one of the real satisfying parts of studying a group like this, is discovering new biodiversity is just intrinsically satisfying. These are, you know, you're looking at a spider here that no scientist had ever seen before. You know, you're the first one to ever recognize it as a new taxon. And so this is one of the new species I found in Panama. And it's an interesting one because I can't figure out what genus it is. Once again, it's definitely a new species. It might be a new genus of Linyphiidae. I don't know. So that's the kind of thing that's really interesting about working with Linyphiidae. So to end up with, I want to talk a little bit about some of the big questions about this group that kind of remain unanswered, that I hope, you know, future generations of arachnologists will answer. And it has to do, who got me really thinking about this is Ingi Agnarsson. He asked me this question. Basically, we were teasing each other about which group that we studied was better. Okay, so this is Ingi Agnarsson. He's a Theridiid expert. He studies cobweb spiders. That's the ones that black widows are cobweb spiders. That actually is not a cobweb spider there. It's a different group, but it's just a press photo. But anyway, he talked to me at a meeting one time, and he was kind of teasing me. He's like, what's so great about Linyphiidae? Theridiids are way better in every way. They're just more interesting. They're a tropical group, like, and so they're really diverse in the tropics, but they're also tiny little web building spiders for the most part. So they're kind of similar. And we went back and forth about this, and it's kind of a dumb question because, of course, any group of organisms is worthy of study, whether it's interesting or not to you. But it really actually was very thought-provoking. It made me think about, well, why am I studying Linyphiidae? Why am I fascinated with them? My first answer was, of course, it's the male carapace. That's why I got interested in him in the first place. And there's more modified male carapaces in Linyphiidae than all the other spiders put together. So that's pretty cool, right? They're little aliens. And he was like, yeah, but it's not really unique to Linyphiidae at all. And not even all Linyphiidae have modified males. And so he pointed out Neospintharis trigonum, which lives in eastern United States, and it's got these two big horns on its head. It's a Theridiid. And then here's one that I caught in Panama. Look at this Dippoena species. It's got this huge, tall carapace. This is another family altogether with a tall carapace. And so really, this is part of what I call the small spider syndrome. When spiders get really small, they can kind of pull off these carapace modifications. They're able to be more heavily armored. And some of them are so heavily armored, they have a shield on their abdomen to protect them. And so some Linyphiidae have those too. No big spiders have those. Well, few big spiders have them because it's just so heavy, you know, when you get that big. And so really, it's kind of a small spider thing and not a Linyphiidae thing. So I went through a bunch of things, and he always had some Theridiid that could do that too. And Theridiids have some things that Linyphiidae don't do, like they're social. There's a bunch of social Theridiids, and we don't have any social. And they've got the black widows. We don't have anything like that. So we did have the island giants. So I told him about the island giants. So that's one cool thing about Linyphiidae, but it's just a few species, right? It's not most of the group. So this is one genus, Orson Welles, was named by my friend Gustavo Hormiga in honor of the larger-than-life, you know, Hollywood director and actor. So these are really big, oversized Linyphiidae too, the biggest Linyphiidae in the world. It's a genus that lives in the Hawaiian archipelago, and these animals are single island endemics. They all each occur on one island in the archipelago. There's 11 of those species was described in this century by Gustavo, and one of them's probably already extinct by the time you described it. Really interesting. You know, most linyphiids are maybe about 2.5 millimeters long, and these start at 6 millimeters and they go to 14 millimeters. So here's one of the, this is the biggest linyphiid I've ever beheld, and that's my hand there. This is Orson Welles' Malus, and that means bad in Latin, and it's named after Orson Welles' movie A Touch of Evil. So cool name. There's their huge web there of a different species. So these are island giants. There's an empty niche there, or maybe there's lack of predators, which allowed these small spiders to evolve into giants, which is really interesting. There's another lineage of linyphiids on the Juan Fernandez Islands, also in the Pacific off of Chile, that are also giants. And so that's pretty cool, but that's not most sheet web spiders. I just had to show you that. I finally came up with two things that I think make Linyphiidae really unique among spider families and kind of really worthy of focus. So here's the first one. This is hard to get your head around, but in my opinion, what's amazing about Linyphiids is what's boring about them. A lot of times people almost feel sorry for me. They're like, why do you study those little spiders? They're tiny and they all look exactly the same. Why don't you study tarantulas or black widows or something? And I say, well, this is what's really ecologically interesting about them. They have this huge diversity, despite apparently being very homogeneous. Other than those modified male carapaces and their genitalia, you know, other than sexual characteristics, their morphology is very similar across the 4,000 species. And their ecology is mostly pretty much the same too. The majority of species are ground dwelling, and they're all using these sheet webs, not all of them, but most of them use sheet webs to capture mostly the same prey, soft-bodied flies and springtails. So that's what's interesting about them, actually, right? The fact that they all look the same. So the high worldwide diversity of these spiders actually, I think, is explainable. I don't think it's really that much of a surprise. It has to do mostly with being small. They're small spiders, and that makes them also very good at dispersing. Their ability to disperse by ballooning is a huge evolutionary advantage for them. Okay, so the small size allows them to evolve quickly, and it makes them intrinsically less vulnerable to extinction. And so over evolutionary time, there's a lot of species that are produced and they don't go extinct. This leaf litter niche is really widespread. It's a huge niche that goes all around the world. And so compared to the size of the spiders, it's just massive. It's like, you know, the deep ocean or something of spiders. And then these animals' ability to colonize isolated and ephemeral habitats is a real advantage too, right? Darwin noticed that linyphiids were ballooning onto the rigging of the Beagle when he was 200 kilometers off the coast of Brazil. And so they clearly are going all the way across the ocean, at least every once in a while. Biologists have documented after the volcanic eruptions in Krakatoa and more recently in Mount St. Helens, the very first animals that were found on those, you know, moonscapes was linyphiid spiders. And so they're really good at doing that, recolonizing habitats. And there's a lot of endemic species on all sorts of isolated habitats around the world. Islands like Hawaii, in caves are isolated habitats, different types of wetlands, you know, in a terrestrial environment, and the tops of mountains all around the world have got endemic species of linyphiids. And so that probably accounts for a lot of this huge diversity of spiders. The thing that's hard to explain, though, is the high site diversity, right? The alpha diversity of linyphiids. You go to one place and you find a whole bunch of species of linyphiids all in the leaf litter, all pretty much seemingly doing the same thing, okay? And so they're somatically, they're very similar. They seem to be about the same size, you know, somewhere between tiny and small, we would say. And so this kind of begs the question about Gauss's principle of competitive exclusion, which says if two species have the same niche, then one of them should be a little bit better at exploiting that niche, and one of them should be able to outcompete the other one and eliminate it. So you shouldn't have all of these species. But yet, here's just one example from my PhD work in South Carolina. I pitfall trapped linyphiids in 8 different habitats, making up kind of a successional gradient from really young habitats, old field and young pines, to 200-year-old riparian old growth woods. And in every habitat over the course of a year, I collected 2,700 adult linyphiids from 55 different species. But every single habitat had between 1 dozen and 2 dozen species of linyphiids. And they're all just from pitfall traps. So they're just all in the leaf litter. They all pretty much doing the same thing. So what's going on there? That's kind of a mystery still. There's some explanations, there's sort of partial explanations like seasonal separation. There's clearly they are adult at different times of the year, and so that can allow them to, to kind of split up the year and kind of coexist. But it's definitely a factor, but probably can't be an all-encompassing explanation. Let me explain. So when I did my PhD research, I found 20 species that were abundant enough to look at their life cycle, and 10 of them were one phenological pattern and 10 were the other. So there's two phenological patterns. Half of them were univoltine, one generation per year, and they were always found in the wintertime in South Carolina. And then the other half of the species, the other 10 species, were found all throughout the year. You could find adults and immatures all throughout the year. And so they probably had multiple generations, and they kind of peak in the growing season in the spring, summer, or fall. And so those are basically the two patterns. And so that can't really explain why there's 24 species living on in one habitat. It's probable also, though, that they're not quite as homogeneous as we think they are, right? Everything looks the same when it's 1 millimeter big and it's 2 meters away from you, right? They all look the same. But when you look at them up close, you realize, oh, there are differences here, and maybe they're not so homogeneous. Linyphiids are often habitat specialists. You find different linyphiids in deciduous versus coniferous forests. You find different species in wet versus dry fields. You find different species in different kinds of wetlands. And on elevational gradients, you find different species as you go uphill. So there's definitely some micro-habitat specialization that's going on. It's not very well understood. There are some that are obvious, like I've been studying this linyphiid called Drapatisca, and pretty much the only place you can find it is on tree trunks. So that's a very specialized habitat that it lives on. And we know about those few species that are myrmecophiles that live in anthills. And then there's quite a number of species that live in vegetation and canopies of forests. This is a species that's an arboreal species in the Pacific Northwest that my colleague Rod Crawford took a picture of. And they tend to have these colored abdomens when they live up in the vegetation. It seems to make them more cryptic. So there is some evidence of microhabitat specialization, but there's also a lot of evidence of ecological equivalency too. For example, a lot of the species that I study are very wide ranging. There's a number of species that you can find in all 48 lower US states. So they must be pretty general to be able to go that far. There's many species that are whole Arctic. They're found all around the world at a particular latitude. There's even some pan-tropical species that can be found in the tropics all around the world. And again, many species can be found in just one small spot, one pitfall trap or 1/10 of a square meter of leaf litter. You'll find a number of species all seemingly doing about the same thing. So this is hard to study, but we really need to look at this more closely to know how important this is. So in the end, I really think that to some extent, I think that linyphiids experience non-competitive conditions in leaf litter, that maybe competitive exclusion doesn't apply. If linyphiid numbers are being kept below their carrying capacity, then they're never going to get abundant enough to actually compete with one another. I think it probably explains a lot of it, right? There's no question there's high mortality and parasitism of linyphiids in leaf litter, and climate can really decimate them quickly, right? If it freezes suddenly, if it gets dry, the linyphiids will go away. If the habitat's flooded, they can drown. And so they're being kept at low abundance. It really makes sense for the system, right? They're tiny predators in this large leaf litter microhabitat. There's a lot of prey availability, but it is spatio-temporally variable, right? So sometimes there's a lot of prey, sometimes it disappears. There's no question high mortality of linyphiids. A couple % of them die every day, usually, in most habitats. They've got all these different things that are killing them. And then linyphiids are known to be very good colonizers in general. Not all of them, but most of them can disperse really well and reproduce really quickly. I documented one species that produced 40 egg sacs. One female produced 40 egg sacs during her lifetime. So they're reproducing really rapidly to make up for high mortality. So this has not been rigorously tested, but I think this non-competitive condition of the leaf litter is probably pretty important part explaining this high alpha diversity. Okay, I know I'm probably running out of time here, but I want to talk about one more thing, and this is really the reason why I wanted to talk about sheet web spiders to you guys in Newfoundland. And that is because of the unique biogeography of linyphiids. This is the second unique linyphiid aspect. I think of them as the rulers, the spider rulers of cool and wet habitats. This clade has an extra tropical diversity peak, which is pretty unusual for a very diverse group of organisms. Now, obviously, there's a lot of groups that have an extra diversity peak. One good example is penguins, right? They definitely do not peak in the tropics, right? But when you get a group that has 4,000, 5,000 species in it, it's very hard to find groups that have such a strong extra diversity peak here, where they're much more diverse at high latitudes and much less diverse actually at low latitudes in the tropics. Their highest diversity is in cool, wet, north temperate latitudes, right about at the latitude of Newfoundland actually is where they reach their highest diversity, about 45, 55 degrees north. They're the dominant spider family, the #1 spider family in terms of numbers of species in all temperate to high latitude habitats. In the tropics, they're found there and they become more diverse and abundant in mountains. So altitude, you get more diversity. So they're among the most poleward of all spiders. And that's a very interesting thing about them. They're found as far north as land goes in the north. And they're found everywhere but Antarctica itself in the southern hemisphere. They're found in these sub-Antarctic islands. They're the southernmost spiders that are known, even though they're also found in all but the driest deserts. They're found in all kinds of habitats, but they really like cool and wet. So this is a Wisconsin peat bog, and the dew in the morning shows up all of these linyphiid webs, and they can become very, very diverse and abundant in temperate, peatlands in Canada, for example. And they also can be adults during the winter in the subnivean space underneath snow. So there's I have an arrow there to show you the little spider there in the subnivean space that forms below the snowpack. And they can be active and even reproduce during the winter in Canada. So I wanted to show you one example of how dominant they are in the far north. This is a study that was done by a late friend of mine, Robin Leach. He studied, this I think is the most northern area that's ever been intensively studied for spiders. Hazen Camp is on Ellesmere Island in what was then Northwest Territories, now Nunavut. This is Canada's 3rd largest island, and it's way up there, right? There's only 115 vascular plants known from this huge island and 1 woody species. And Robin collected 20,000 spider specimens over 2 years, so very intensive. And yet he only found 13 species of spiders from only 4 spider families out of 128 or so that occur. So these are the most northern spiders of all, and almost 70% of them, most of the species, are linyphiids. So linyphiids are really the northern spider. This is just one example of one of the species he caught, and it shows his map showing the whole Arctic distribution of Vulcaneria karpinskii, which is found, throughout Europe and Scandinavia, but also found throughout Siberia and Canada, Greenland, and all the way down into the United States, even farther south in Green Bay. There's Ellesmere Island up there. So that's a pretty common distribution pattern for linyphiids. So this table here is showing how powerful this extra tropical center of diversity is for linyphiids. So I looked at places that had really well-known species lists, relatively speaking. So we'll start up in the far north in Iceland. Ingi Agnarsson studied the spiders of Iceland in the 90s, and he found 91 species of spiders. 66 of them were linyphiids. So that's over 70% of all the spiders in Iceland are linyphiids. Here's Newfoundland. So Dondale and Pickavance in 2005 came out with a nice paper that documents over 350 spider species on Newfoundland, and over half of them are linyphiids. So you've got a lot of linyphiids on the island, undoubtedly ecologically important. If you look at Canada itself, there's almost 1,500 species known in Canada, and over 500 of those, about 38%, are linyphiids. Down in Wisconsin, there's fewer. There's about 20%, a little more than 20% of all the species, so almost 500 species and over 100 linyphiids. Texas, you start to see a drop off, right? So there's 3/4 as many linyphiid species, but there's twice as many spider species in Texas as in Wisconsin. And so they only make up less than 7% of all the species in Texas. It's probably an underestimate, but you get the idea. Panama, there's definitely more than 29 species of spiders, Linyphiids in Panama, but there's also way more than 1,200 spiders too. So right now they make up about 2.4% of the Linyphiids of the spiders in Panama. So you can see this is a very strong pattern of, being more diverse in cool and wet areas. If you look at the zoogeography of the entire family worldwide, this is work of Andrei Tanasevich, a Russian linyphiidologist, and he is documenting that over half of all the species in the world are found in the Palearctic, and then about 20% are found in the Nearctic, right? Canada, the United States, and Mexico. And then it really starts to drop off in other regions of the world. So they're found everywhere in the world, but they're much more diverse in the Northern Hemisphere, and particularly the Palearctic. That seems to be the center of diversity. And there's two things going on here. There's no question that Europe is better studied than any other place. So they found all their Linyphiids pretty much in Europe. But when you look at where the Linyphiids are, there's actually more species of Linyphiids in northeastern Asia, basically Siberia, than any other place on the planet. So that is the center of diversity of Linyphiids as far as we can tell, which is very interesting. That is an unusual situation. So that also begs this question, well, why are they? What is it about Linyphiids that makes them so good at high latitudes? Why are they the rulers of the cold and wet? And a lot of things get kind of thrown out right away when you start looking at these things. If you look at the physiology, you think, oh, maybe they're super winter adapted. But from what we know about them, they have basically the same physiological adaptations that other spiders, that Robin found up in Ellesmere Island have. So other wolf spiders and things, they all seem to use antifreeze compounds to depress their freezing points so they don't freeze, until you get really cold. And they all seem to overwinter as immatures or adults in protected microsites under the snow or under rocks or whatever. And their eggs are not cold adapted and will readily freeze. So that's pretty much the same for all spiders. The prey, it might have something to do with it, but the main prey, springtails and small flies, are found everywhere, right? They're quite abundant in Georgia, right? So why wouldn't there be a lot of Linyphiids down there? It is true that they're maybe even more abundant in cool, wet habitats, springtails and small flies, but I doubt this is a real explanation for why they're so very diverse in the north. And then morphology is something you can, if you're a Linyphiid person, you can discount this right away. It would be almost impossible for, I could give a Linyphiid person a Linyphiid and they wouldn't be able to tell whether it was from Panama or Iceland by looking at it. No way. They have basically the same morphology in wet, cold areas as in hot, dry areas. And then Linyphiids themselves are not morphologically different from other species either. In fact, it's very difficult to identify, Linyphiids compared to other web-spinning small spiders. So there doesn't seem to be anything about their morphology that's super distinctive or special. What I think it might be super interesting about them is this web, this sheet web. I'm not the first person to suggest this, but it's possible that this web works particularly well in rainy conditions. It's relatively strong. It's a semi-permanent structure. It does not rely on stickiness of silk to catch prey. It relies on the knockdown ability of the scaffolding, and it relies on the tangling ability of the silk. And so it can work even when it's soaking wet. It might also act as a rain cover, right? Because most of the time Linyphiids are hanging up down at the bottom of the web, upside down underneath it, so they may be protected from the weather that way too. As far as I can tell, this has not really been studied in any great detail. So the functional biology of Linyphiid webs is one thing that I think is a really neglected topic in biology. It's much easier to study larger webs with fewer, thicker lines and simpler structure. And there's huge numbers of papers about orb weaving spiders, but very few papers about sheet web spiders. Okay, so that's it. I want to acknowledge my mentors. I've already showed you Dak and Don, but here is the curator of spiders at the Field Museum. Petra Sierwald is definitely a collaborator, but really a mentor of mine. And then I would like to acknowledge my Linyphiid buddies who are actually here today, Mark and Nina. This can be a really lonely field. You're so specialized. But if you have people to work on these problems with you, it makes it a lot more fun. So there we are at the Field Museum a year ago in March, trying to identify some spiders, mostly of mine. So thanks. And then finally, I'd like to acknowledge the contributions of my wife and the support of my wife, who actually understands, what it is I do. She's also a PhD ecologist and has been very supportive of me over the years. So thanks, Vicki. Okay, that's it. I don't know if we have any more time, but I'd definitely be happy to talk more about Linyphiidae if you want. Should I?
That was awesome, Mike.
Thanks.
Yeah, if people have questions, they can just type them. Just say you have a question in the chat box, and we can get the question period rolling.
Maybe I'll keep it on the slideshow in case someone wants to look at one of the slides. But
yeah, sure. Yeah, Adam has a question. If you want to go ahead, Adam.
Hi there. Thanks so much for the great talk.
Welcome.
So super interesting to see the diversity of morphology and mating strategies and genital structures and like the seasonality of mating.
Yeah.
But with all that alpha diversity, I'm curious how common are hybrids, and how do you deal with hybrids as a taxonomist?
Yeah, we don't really know. I think we make real assumptions by looking at large series of animals. It's not really very common that we find sort of intermediate looking spiders. And that, I gave you that one example of one with some intermediate forms, and there definitely are some, but I would say that they're in the minority. There's probably more hybridization and more cryptic species than we're aware of. So as we learn more about, molecular tools, and we use molecular tools, there's no doubt, I think we're going to find more, not so much hybridization, but cryptic species complexes where what we think is one species is really 10 species. I think there's going to be a lot of that. But there doesn't seem to be a lot of morphological evidence of a great deal of hybridization, although it does sometimes happen.
And I guess it's tough anyway if you've got like so much intraspecific variation in morphologies to start like identifying intermediates when there's already so much like within species variation. It's got to be so tough without a molecular tool.
That's true. Although the genitalia have a tendency to be very, they're under a lot of selection to be a particular shape. And there oftentimes is not very much difference between individuals in the genitalia, even though there might be quite a bit of difference in the coloration and things like that. And so in general, I'm pretty confident that there's not like wild hybridization going on in a lot of these species. But there's no question that we're already wrong a lot about how many species there are. When you really start studying them, they tend to be, cryptic species tend to be much more common than we used to think. So it's probably a lot more than 4,000.
Thanks very much.
Welcome.
I guess I'll go with the next question.
Sure.
So, I mean, just going off that, are you guys down to species? Are you guys using a morphological like species concept? What kind of species concept do arachnidologists use?
Yeah, absolutely. We're, in theory, we believe that we're looking at we're interested in reproductive isolation. And so that, that's what we want, is to have reproductively isolated units be our species. But honestly, how often does that get tested? When it does get tested, which certainly has happened many dozens of times in spiders, it tends to really link up with the morphology. So it's not that common to find, this kind of hidden, species, unless they're just, cryptic species. But in terms of, it seems like the morphology, if you see two different palps, probably it's going to be two different species. But honestly, what we're doing is essentially making assumptions. Most of the time, no one has ever seen these species alive before. Most of the species that I work with, nobody's ever seen them alive. No one's ever seen them mate. And certainly no one's ever tested, make sure that the females and males are actually mating and can only produce egg sacs. So there's no question there that it's a tricky thing, that we're making assumptions that we hope are true. The morphology seems to back it up in general, but we're wrong a lot too.
Cool, right on. Yeah, it sounds like Kier has a question.
Oh yeah, Kier.
Yeah, thanks. Wow, awesome talk. I'm curious about the bog dwelling spiders and how specialist they are. Are they widespread across the boreal regions, or are they widespread across like North America, for example?
Yeah, I wish I knew the answer better than I do. I mean, there are some species that you can find in wetlands that are generalists. Those webs that you saw were mostly something called Frontinella pyramitella, and it's found like in my backyard, and it's found in New Mexico and Maine. But a lot of the species that you find in especially peat wetlands, you know, a really distinctive wetland habitat like that, are really limited to wetlands. Like you cannot find this entire genus called Saddle Atlas. I don't think anyone's ever found one outside of a wetland. They're very restricted. And I guess there's a general rule that as you go farther north, they're more likely to be whole Arctic. And as you go farther south, they're less likely to be whole Arctic. But other than that, so there are certainly a number of Canadian species that are whole Arctic and are found. A lot of them are probably also the ones that you find in Russia. But when you go to Siberia, they've got a whole other set of species too. So it's kind of a partial separation there.
Cool. Thanks.
Cool. I think, sorry, I don't know the first name for this next person, but last name is Sandlin.
Oh, Nina.
Hey.
Nina.
Hey, Mike. So as an ecologist who turned into a spider researcher, when you're thinking about the web as what sets them apart, can you begin to imagine a practical experiment that could be done to test that hypothesis?
Yeah, I can definitely imagine it. And I've seen a lot of talks at arachnology meetings, and I look at their talk and I go, I wish they would do that with Linyphiidae. So there's a lot of people that do not just behavioral work, but sort of physics work on silk and webs, and they kind of define different parts of the web, and they can understand the tension, and they can measure the stickiness. And all of this stuff is like, you could really understand the ecology much better if you could document which species make what kind of webs and document something about their physical properties. That is not work that I'm going to do, right? I didn't even take physics in college. I would not be the person to do that. But you can see that people are doing this kind of work with the orb web spiders. And at some point, the sheet web spiders have got this huge range of different kinds of sheet webs and so many species. And there's a lot of cool questions out there. If somebody would, you know, it's going to be hard to do. They're hard to find. As you guys know, it's hard to find a sheet web without even destroying it. And so it's that taxonomic impediment essentially too. You find a little spider on a web, and unless you capture the spider, kill it, and take it back to the lab, you probably won't know what it is. So that's what a lot of the behavior actually has done, is you watch the behavior, then you kill it and see what it was one time. And so it's hard to study such small animals the way I'd want. That's why we don't know much about Linyphiid biology, I'd say. Thanks, Nina.
Hey.
Good question. Michaela, go for it.
All right. Well, as a lot of other people have stated, that was extremely interesting, and also just to learn more about how it comes from like a taxonomical point of view, because that's not often something you hear. Like, you don't often hear of people whose job is working on taxonomics. And so within that, like, how difficult is it to classify new species of Linyphiid or spider in general? And like, do you think that's part of the reason there's not many that have been fully documented, and why like Panama has such a low number?
Yeah, that's a good question. It's hard to answer. I guess I would say that it's easy to find new species of Linyphiids. What's hard is to actually know that it's a new species. That's what's hard, because this is a group that's poorly enough known that you can't just go to a book. You know, I maybe made it sound like that, like you could just go to a book and see what species it is. But in many cases, Nina and Mark and I are using literature from the 1920s, 1930s, and very poorly, by today's standards, they were poorly drawn, poorly illustrated, and that's what we're using. That's all that people know about these species. So trying to even figure out whether it is a new species or not is difficult, and that gets even more difficult when you go to where the new species are. So if you're doing work in Wisconsin, you can often recognize this must be a new species because I can't identify it. But when you go to Panama, you can't tell whether it might be quite well known in Brazil but you just don't have the knowledge, and you don't have this rare Portuguese paper, that is hard to find. And so, just trying to find what it is definitely tricky. And then, of course, after that, there's other problems. The taxonomy is not well known. There's a lot of species, and they are small spiders. They're small enough that they're kind of at the border between compound microscope, where you put things on a microscope slide, and a dissecting scope. So the vast majority of the work we do it on a dissecting scope. But really, a structure that's 1/10 of a millimeter long, that's too small to see well with most dissecting scopes. You have to get a really good dissecting scope. You really should put it on a microscope slide, but these are soft, squishy, three-dimensional animals, and you can't just throw it on a microscope slide. You'll squash it. And so we're right at the border between those two types of microscopes, and that's another thing that makes it difficult to work with this group of spiders, or to work with small spiders in general. Thanks, Michaela.
Thank you.
Cool. I actually have another question to ask. I guess I'm kind of interested in what value the leaf litter has as a place to build a web for Linyphiids. Like what role does it fill for their environmental niche?
Yeah, that's a good question. One thing about the Linyphiids that I've reared a bunch of Linyphiid species, trying to figure out what their life cycle is, so I raised them from an egg all the way through when they're making their own eggs. And one thing you can say about most leaf litter spiders is they really dry up fast. They're really susceptible to desiccation. And that makes perfect sense because they're very tiny animals. So they've got a lot of surface area and just a little bit of volume. And so that's a really important thing about the leaf litter, is that it's usually much moister and has much higher humidity than the rest of the ecosystem, that terrestrial ecosystem. And it actually kind of begs other questions, right? How can these vegetation dwelling spiders survive without drying up? But certainly the leaf litter is very important just to maintain conditions near 100% humidity. And then after that, I would say it's a place where a lot of the animal biomass exists in a forest. You look at the forest and it seems like it's mostly trees, but a large chunk of the animal biomass is right in those couple centimeters at the very top of the soil. And so that's where a lot of the food is too. Now, it's a dangerous place too. That's where a lot of predators are. And there's no question Linyphiids get eaten by everybody. And but I'd say that's probably why they're so diverse there, is because there's so much food. It's a really productive, in terms of secondary production, it's a very productive part of the ecosystem.
Cool, thanks.
Yeah, they're kind of an important part of the brown food web, we call it. The green food web is the one people think of, right? The plants produce the food, and then it goes to herbivores, and then it goes to predators. But a lot of plants actually never get eaten. They turn into leaf litter and sticks and stuff. And so there's a brown food web that has centered on microbes and fungi, and then springtails and things like that eat it. And spiders are actually very important in getting food from the brown food web and making it available to the green food web, right? So it's transferring energy to toads and salamanders and birds and things like that, that otherwise would be trapped in this microbial loop, right, down at the bottom of the soil ecosystem. So I think that's maybe the main argument you could make for why Linyphiids are ecologically important, other than just diversity.
Cool, perfect.
Thank you.
Katrine has a question.
Hi, thanks again. That was a great talk.
You're welcome.
Just kind of thinking about the diversity of places that you're finding these spiders. One thing that jumped out to me was the vegetation. Not only are the species different, but the structure of that vegetation must be very different.
Yes.
And do you think there's some kind of relationship between the structure of that web and the ability of those spiders to kind of, kind of establish themselves across such a diverse vegetational plane?
Yeah, that's a great question. And it sounds like you're an arachnologist, because that's one thing you start to discover a lot when you think about spiders and where they are. Like, spiders don't seem to care what species of plant they're on. They don't know anything about botany, but they actually can be very picky about plants, but they care about the structure. And so it doesn't matter what species it is, but some spiders really prefer a particular kind of plant, or maybe like some palm trees actually trap a litter that's set above the floodplain so it stays dry. that's super important for the spiders. And so spiders really do, I think they separate across the ecosystem by having different places where they make their webs. And those of us that collect spiders, we know, oh, if you want this kind of spider, you do beating in this kind of plant and you'll probably find them. And so there's no question there's an architecture of the vegetation that's very important that the spiders respond to. And so some of them need to have certain kinds of density of branches and so on in order to make their webs. And yeah, that's an important thing. It's hard to study. That's another thing that's hard to study. It's hard to quantify, you know, how the spider sees vegetation. So we oftentimes are looking at things like a peat bog or this species of tree as like a surrogate for that. That's not really what the spider cares about. It's responding to the environment in a way that we haven't figured out how to measure yet, really.
Cool, thank you.
Yeah, it was a great question.
Cool, Levi, you want to ask a question?
Yeah, hope there's still time. Yeah, thanks for the talk. That was super interesting and kind of a lot different than some of the talks we've seen recently. So it was really nice to see. I was curious about the relationship between like if I'm understanding correctly, like you think that the Linyphiids might respond to like the amount of precipitation.
Yeah.
And that like has something to do with their web. But there's I don't know. I mean I don't know. Maybe there's some correlation between like latitude and precipitation, but is there like, can you elaborate on that more? Like is it more to do with like the temperature and precipitation or like that kind of precipitation that?
Yeah, that's a good question, and it's more complicated than I made it sound in the talk. There's no doubt. And when you think about the tropics, that's when the whole argument starts to fall apart a little bit, right? Because you can find Linyphiids, you know, in the lowlands in the tropics, but they're not super abundant. There's other spiders that sort of take over, and they're not as diverse. And then when you go into the mountains in the tropics, they start to dominate, just like they do in the north. And so, you know, if you think about the precipitation, it's a little complicated there because they're getting a lot of precipitation in the lowlands. The precipitation does increase with altitude, but who knows? Maybe it's more about the cool temperatures in that case. So it's confounded by a lot of things, and I'm not pretending to be able to understand it. I don't think anyone knows exactly why they respond the way they do, but there's a very strong pattern though that when you get into cool, moist habitats tend to have a lot of Linyphiid species. When it gets really dry, Linyphiids really drop off. So that's certainly important in their biology. They're not well adapted in general to dry areas. Although you can find them I think I mentioned, you can find them in the Sahara Desert, up in the Atlas Mountains. And they're actually quite diverse in the Southwest, in the desert Southwest, New Mexico, Texas, Arizona, but they're just not very abundant. They come out, you know, in the wintertime when there's a little bit of moisture, and then they disappear again. So I'm not going to even pretend to know what's going on there, but there's a complicated relationship between precipitation and temperature and who knows what all.
That's pretty neat. Thanks.
Yeah, thank you. I wish I could tell you more precisely what was going on.
Are you strapped for time, Mike, or are you doing good?
I am not, and I appreciate you guys being so patient. I'm sure I went over an hour there on my talk.
Yeah,
so
that's good.
Great audience.
I guess I have another question. I mean, it seems like you have a lot of work in queue, like you have a lot of backlog data. So what keeps driving you to travel to different areas, and is it to bring younger like researchers with you to help inspire them for grad school in this field? Or
that's an awesome question, and it kind of goes back to that taxonomic impediment and some of the real frustrations of studying Linyphiids. And I think my two colleagues here could definitely relate to when I say this. I've got like a lifetime's worth of work already in bottles, you know, in my lab. And that's a real problem, really. The back end of the work is very slow, and it seems like it never gets done. And then people are always wanting you to go and do a new thing. It's, you know, the granting agencies want you to go and do a new project, and people want you to get involved with their project. And so I'm always working on these new projects, and I've got these old projects that just never seem to get finished. And it is very frustrating. And I have to say the reason why I still do field work is because that's kind of the fun part, right? The reason why I got into field biology was because I love to be outdoors and I like to see the living animals in their environment. Although really, you know, I could probably just sit and study dead animals, you know, until my last day and never run out. So it's kind of like almost for me that I'm doing it, but it is true that I'm increasingly interested in helping, you know, young people get into arachnology and get into ecology. And so taking students on these trips is becoming more important to me. And it's no longer so important that we're catching animals that I'm going to publish. It's more important that I want to show them how awesome, you know, these animals are and these places are.
Yeah, perfect.
I will say, if it wasn't for, I went on one of his Panama trips, and if it wasn't for that trip, I like before that trip, I was like afraid of spiders, would never get close to them. And that trip definitely like opened my eyes to spiders. And like now I'm working with like pitfall traps and I've got like spiders in my lab right now. And so I will say like, you want to feel that it's hands-on with them. And if it wasn't for that, I don't think I would be even close to touching one.
Thanks, Michaela. I really appreciate that. And I think that's a good thing to say too, is that when you're an arachnologist, a lot of what you do is you deal with the public. And these are animals that have a real cultural association, right? Everybody knows about spiders and most people fear and loathe them. I meet people all the time that are paralyzed with fear. And I also meet people all the time that wish that they were all dead and they want to kill them all with fire, right? And so that's something that's a really important part of my career. I want to lessen people's fear and I want to lessen people's hatred, to be honest with you, of a lot of invertebrates. So that's something that I spend time doing. And I spend a lot of time on Facebook identifying bugs and stuff. And I don't need to do that, right? That's not something I need to do, but I really enjoy kind of that outreach. And I think if you're working on invertebrates, that's a responsibility that you have, is to try to increase people's understanding and decrease people's fear of these mostly completely harmless animals. So thanks for going out there and preaching the preaching the good word. They really are harmless and very, very important parts of ecosystems, and also just an important part of our world, right? They're part of what makes our world different from any other corner of the universe. So.
Great.
All right. Well, thanks so much, everybody. I really appreciate time you took, and you were a great audience. Really.
Yeah, we appreciate you coming in. Yeah. Yeah, we always try to diversify the talks as much here, and it's always good getting other fields.
Yeah.
Yeah.
Right.
So we're very happy that you came.
Definitely not fish physiology, was it?
No. Very far from that.
All right. Well, good luck with your studies.
Yeah. Yeah. Cool.
Thank you.
Yeah. Thanks for coming, Mike. And
sure.
Yeah, we'll end the meeting here if everyone's good with that.
Okay. Yep.
Thank you so much.
Thanks, Mark and Nina. Thanks for coming. Bye-bye.
All right. Bye.